Yeah.
It should
Yeah, you started the process.
start a broadcast. This thing is on presenter view
Screen two?
and screen two. Yeah.
Okay, yeah.
Except it's not going through.
Send it.
So guys, once you're done, just put it in the
You know
envelope.
what? I just need to get into the keyboard. I just need to
I
activate.
think it's not this one. It's
Okay, it's dock camera. Then that's fine. I can do dock camera too.
No, it's just
is this one. Is it?
it's sharing now.
I can do it off of either
Okay,
one. It's just
okay.
going up there.
Yeah, sure. Any feedback? To you? Yeah. To you.
Yes, I mean, I think it's connected to this computer.
I don't want to ruin it. No, you didn't include it. Should we do escape? Or we set it up? Can I do that? You know where it is? Like the other one. Where did he go? It's on Facebook.
I don't think we need to.
Yeah, they know who we are.
Yeah, like I was kind
I
of
was
like
surprised when
surprised.
you
Yeah,
said like they didn't know our names.
yeah. I was like, I think they know. I was like, well, at least I'm not worried for me. Like, I'm the guy. Like, maybe they'll mix you guys up somehow.
I don't think so. Everyone
Maybe.
knows my name. First name. Vanessa.
Yeah,
Last
maybe.
name they don't. Why that's like
Man, I'm gonna get no feedback. Sucks. Job like 10 students. Max. Max. You're lucky. I'm gonna grill them on Monday.
I haven't counted mine. 5,
Like
6.
y'all have to send me email feedback now. So now I know who said what. They're gonna be like, oh.
9. I think I come across as a lazy TA.
That's at least what?
A lazy TA. I don't
Why?
think so. Yeah,
Probably me.
I think I come across like that. I don't think you do. Because you talk to them a lot. So I say a lot of I don't know.
But I don't think it means I'm lazy. I think it just means I'm like,
Chill.
yeah, I'm
Wait,
chill. I'll
is
be
this
like,
No.
yeah, it's on.
No one ever repeat what you just said. Hey, can they hear it? I don't know.
It's
I don't
made for
think
that.
Just because it's
Yeah,
video.
no. Just mute it.
Okay. So fancy. Some privacy for the team.
Yeah. Excuse me.
I feel like they didn't all take one away. Like my students, I see them chatting. They're not feeling good.
You call it.
What?
And I was like, Dr. Q. I'm one of.
Did you have a lecturing on Friday? Or is there no time for me anymore?
So what's going to happen is Monday we're getting into precision medicine. So if you want to come in and talk about biology, talk about
Why do genetic testing? I could have 25 minutes. Oh wow. Okay. So Monday? Okay, so Monday? Okay, I'll send you the slides ahead of time with the script inside them. At least the slides. Okay. No, it's good. Time flies when you start to.
That's true. Like we had the presentation for once and it was 2 hours. Wow.
Get to the biology.
You guys can't take this. So make sure if you take it back to the HMB office, you sealed it and then you write like HMB204 because they're going to get a lot of these and they're going to mix them all up. So
How long are we giving them to
um,
participate in the survey?
you can.
I muted it because we were standing and talking.
No,
How
you
long?
didn't.
What?
How long should we give them? Like,
You
are we
can
gonna
just-
make another announcement?
Yeah, you can just tell them next few minutes.
Okay. Hey guys, if you haven't already, please take a survey from the front and a coffee or tea. And we'll give you a few more minutes to fill it up for us. Thanks. Oh, we muted the wrong one before.
Yes. Can you just also mention they'll be getting their assignments back in tutorial on Monday?
Okay,
So it's useful to actually show up in case they're thinking of not to.
sure. Another announcement, guys. You'll actually be getting your assignments for your presentations and your marks and everything next tutorial, so on Monday. So you'll be doing a review session for your final exam and getting your assignments back. So it's a good idea to come to tutorial Monday. Your last tutorial, okay? Yeah. I feel like saying, P.S. I'll be lecturing Monday for sure. You can do it. Also, I'll be giving a short lecture on Monday. I'll be giving a short lecture on Monday. Alright, are we supposed to stay?
I think if there's no you take it, give it to Dana, and make sure that you've written 204. Okay, well, good. Best of luck to all of you.
I have a committee meeting today. No. Chapter. No, she did. Dr. G, where's my good luck?
It's okay.
Maybe it's one of them.
It's okay.
Could you ask if there's any more? Any other surveys? Is anyone still working on their
Is that better?
survey?
Sure.
No? Okay, cool. All right, see you guys next Monday.
Okay, thanks for taking the time to do that. It's really great that you are providing feedback to the TAs. As you know, most of them are TAing for the very first time in this course. And some of them were my former students. So one day, if you're thinking about it, down the road, maybe in 2 or 3 years, maybe you'll be a TA back in this course as well. So that's a really cool objective to be working towards. We're about to head into sort of the last bits of the course. So there are still important things that we need to chat about and go through related to this course for your final exam and to make sure that you're ready for your final exam. But I also wanted to give you a good idea about precision medicine, why we're interested in what's known as precision medicine. Some of you might have heard of it as personalized medicine. Some of you may not even care about personalized medicine or precision medicine, but I can guarantee you that in your lifetime, probably not so much in my lifetime, but in your lifetime, all of you actually will be affected by precision medicine, personalized genomics, finding out your own pharmacological genetic background. All of these things actually will impact you in the next 5 to 10 years. When you're off having your own families, when you're doing your jobs and everything else, these are the types of things that are going to affect you. And so today, we're going to start looking at some of the things that we've previously looked at in this course, things related to BDNF, which I know we've chatted a little bit about, and then single nucleotide polymorphisms as an adjunct to BDNF, and then get back into this discussion about why these different polymorphisms, why are we interested in our genomes? Like, why do you even care? Diseases notwithstanding, like, even if you don't have a disease, should you care about your genomics? And I would argue that you absolutely should, even if you don't have any disorder, any disease, any genetic issues whatsoever. And so I'm going to start off with BDNF. I'm going to talk to you about depression or major depressive disorders today, and then I'm going to talk to you about why genetics is important. And then we're going to continue this discussion on Monday, and then Friday we're going to have a full review to make sure that you're ready for the final exam on the following Tuesday. So I'm going to go back and talk to you about the BDNF system. You don't have to worry. I'm not going to ask you to go back to your old slides. I'm hoping that most of it is somewhat familiar for you. I'm not going to talk to you about schizophrenia today. I'm going to come back and talk to you about it maybe a little bit on Monday, and I'll talk to you more about major depression. And I have to apologize. There are some aspects of this slide that are still from the previous lecture. So I'm going to talk to you about BDNF. And many of you will know that BDNF, this brain-derived neurotrophic factor, is really important for brain health. Do you guys remember that? You need BDNF to keep your brain healthy. And that's somewhat of a universal sort of statement that every neurobiologist makes. That BDNF is good for you. That you need more. The more BDNF that you have, the better your brains are. The more BDNF that you have, the smarter you're going to be, et cetera, et cetera. It's all a lie. None of that is actually true. Now I'm going to tell you the reasons why, as a neurobiologist, why some of this is actually not true. The way that your brain learns is that different nerve impulses go from neuron A to neuron B. And if we strengthen the connections, the communication, the neurotransmitter responses between neuron A and neuron B, we say that you've learned something. The synapse has gotten stronger. We say that it has potentiated or undergone what's known as long-term potentiation. We know that one of the molecules that makes a small signal, like, I'm not going to remember most of your lecture today, Dr. Chu. In fact, I'm going to forget about 75% of it as soon as I leave the classroom. The 25% that remains is because some of your synapses have become a little bit stronger. And the synapses that have become stronger aren't like the ones up here. They're the ones that have somehow caused the release of a little bit more BDNF and caused this particular group of synapses to retain that information a little bit more. And one of the things that we know that is important is that these AMPA receptors, or these AMPA-Rs, at these synapses are really an important part of developing this stronger signal. This means that from neuron A, that you're hearing this information from, going into your hippocampus, if you're going to remember it, it has to release enough BDNF for you to remember. That BDNF also has to go across and interact with the TrkB receptors that we chatted about before, influence the stability of AMPA receptors, and then you remember things. Okay, and that's, in a nutshell, one of the ways in which we remember. We need BDNF. And again, we've talked about this many times. Exercise, which you should be studying for your final exam. We talked about BDNF. We talked about BDNF being important for you overall in brain health. All of that, again, in theory is correct, but the reality is that it's not maybe as good as we'd like to say. We know that another thing that is important in this pathway is that it can't just be a one-way communication, that a molecule like neuronal nitric oxide synthetase has to be involved and cause nitric oxide to be retrogradely, a gas, nitric oxide to be retrogradely sent back to also strengthen this BDNF signal. So we know a lot about this BDNF, but the question at the bottom is really an important one. Is all BDNF good for you, right? I've been telling you that in the exercise module. We've been talking about with the TrkB and the NGF neurotrophin P75. I mentioned that as well. And I'm telling you again, when you're learning, BDNF has to be there at the synapse for you to learn well. But is all BDNF good? It turns out that we have, generally speaking, a polymorphism in the BDNF gene. And at position 66, You can either have a methionine at position 66 or you can have a valine at position 66. I can tell you that everyone here in this room actually has one polymorphism. I can tell you that with almost like 99%, without even genotyping you, I can tell you that it's about 99% accurate, that you have, all of you have the same polymorphism, and that you're all likely going to be homozygous for this. Depending on the polymorphism that I'm going to get to in some more detail, whether you have one version or the other, that is a polymorphism at one particular genetic locus, you are going to either increase or decrease the secretion of BDNF. Okay, so this is weird. Like, we keep hearing about BDNF, but if you have one isoform of BDNF, even if you release it, it's not as good. It just doesn't get released as frequently. And so there is a fundamental difference in the type of BDNF that you have. So if you have one isoform, for example, the methionine 66 version, you're going to have decreased secretion and release of BDNF. That means that your brain might be smaller. It might mean that you have less capacity to remember things. Your memory is not going to be as good as someone who has the 66 valine isoform of this. You might actually make yourself more susceptible to anxiety. You might make yourself more susceptible to depression, a whole host of factors. So this one single polymorphism affects learning, memory, as well as mood overall in your brain. We know, for example, that BDNF is important, and I've mentioned things in this course before, that if you're looking at normal BDNF signaling and you block BDNF binding to TrkB receptors using TrkB antibodies or anti-TrkB antibodies, the number of synapses is going to go down. You're going to be reducing the increases that you see after exercise in terms of memory enhancement, etc. So we know that BDNF is indeed important, but we also know now, in retrospect, that the type of BDNF is equally important. So especially when we talk about the area of the brain known as the hippocampus, I want you to be thinking a little bit about this. One of the reasons that we know a little bit about BDNF is actually from a number of different studies. And I actually hand chose these different studies. That there's a link between the amount of neurotrophins that you have, synaptic plasticity, and the good genes that you turn on to make your brain bigger, to reduce anxiety, etc. So Lou and Gottschalk started this whole conversation about more neurotrophins, better brains. BDNF is a neurotrophin, a better brain. Pu then carried this on in 2001, and in 2016 you get Ju talking about all of this. So you get Lou, Pu, and Ju all in the same sort of sentence. So if you're thinking about it, neurotrophins, generally speaking, are a pretty good thing. However, after the Human Genome Project was finished, and we knew that there were certain genes that were important in terms of our brains, and one of the most important genes that people wanted to go back and examine was BDNF, they realized that in the human genome, there are actually frequent polymorphisms in BDNF, particularly at position 66, which causes a conversion of the normal amino acid at position 66 from a valine to methionine or vice versa. So people knew, for example, that different individuals that have psychiatric disorders like schizophrenia, it's not something that is acquired. We know that there's a strong genetic component. There's about 1 in 100 individuals that will have schizophrenia. It is something that is genetically predisposed. That if you took a look at their genome, there was a strong suspicion that something's wrong with their brain. Since we know that BDNF plays a huge role in brain development, normal synaptic plasticity, learning, memory, anxiety, depression, all of that, there were strong suspicions back in the early 2000s that this gene, BDNF, was not working well in individuals with schizophrenia. There was a huge race on, because once all of this genetic data that was released in the year 2000 and 2001, and people had free access to the Human Genome Project, and people saw that there were genetic variations in BDNF, according to the Human Genome Project data, there were two different groups that were racing literally to try to figure out, do schizophrenics have an abnormal version of this particular BDNF gene? It might make sense. Schizophrenics have all sorts of issues related to cognition. They're not really good thinkers. They can't really make good judgment calls. They also have really poor memory. They have lots of things going on in their brains. And if BDNF is important, you would suspect that maybe they have one of these different isoforms that I mentioned to you before. Now, I put huge here in quotations, because at the time, in the early 2000s, if you could get 600 individuals and genotype 600 individuals, that was a lot of data points. Nowadays, we look at 600 and it's laughable. We're looking at tens of thousands of individuals at a time in terms of our data points. But at the time in 2003, when Egan was trying to publish, this was actually considered a large number of subjects. Now, this is a really busy slide, but this is a really important slide. I'm going to take you through it twice because I want you to understand the data that is being generated from Egan's paper in 2003 in Cell. And this is actually one of the really cool slides that he has, because this is actually a data slide that doesn't actually show you any data. In fact, the data that they wanted you to see first is down here at the bottom. Remember, there was a hypothesis. Their hypothesis was that if you had one variant or another of BDNF, that it would be something that would be carried over to schizophrenics. It would probably be implicated in a not so great brain. And so they wanted to know, if we go back and we genotype individuals that have clinical features of schizophrenia, do we actually see them not performing well? So they do a memory test. They give them a psychological memory test. Then they go back and they genotype them, and they wanted to know, if you have one genotype versus the other, do you actually perform better in a memory test? Because remember, learning, memory, BDNF release, synaptic plasticity, strengthening of connections. Schizophrenia, it's messed up. They have poor memory, poor everything else going on in their brain. And one of the things that when they looked at the BDNF genotype, one of the things that they saw was, if you are diagnosed with schizophrenia, which is the patient group down here at the bottom, do you see any difference between any of the genotypes? If you have valine, valine, valine methionine, or methionine, methionine, if you're homozygous or heterozygous, do you see any difference in memory performance? It's either yes or no. Whoever's saying maybe, that's a good sign of independence, but not the right answer here. Sometimes it doesn't pay to be stubborn. So there's no memory difference. That was their hypothesis. Their original hypothesis was, if you have schizophrenia, we should see clear differences in memory. But of course, we know that anyone who's diagnosed with schizophrenia is not going to be doing well on a memory task. Their experimental design from the beginning was flawed. However, here's the saving grace. They actually did a control, and their control was this. The control was to get other individuals, siblings, their own sort of brothers and sisters who are not diagnosed with schizophrenia, and have them genotyped and have them do the same memory task, and just any old individuals that were age matched in the general population. And these were the sort of controls that you see at the top. Now, when they did that, where they were not expecting to see any differences, just in the average person, no schizophrenia whatsoever, one of the things that they observed is there's a clear difference. They now start to see that if you have the valine form of BDNF at position 66, then you are performing much better on a memory task than someone who is homozygous with methionine at position 66. So you can't tell in schizophrenia, because there's all sorts of things wrong in a schizophrenic brain. But in a normal brain, when you go back and check, if you have one isoform, the valine isoform here on the left, you're always going to do better on a memory task. That was surprising to them. It wasn't originally their intent to check the general population, but this genetic analysis actually strongly implicated the fact that when you look at these hippocampal dependent forms of memory, and in the hippocampus, we have lots of BDNF, and in the hippocampus, this is where learning and memory occurs. And in the hippocampus, we know that there are mechanisms like BDNF, TrkB, AMPA receptor stabilization, and nNOS that occur. So if your BDNF isn't getting out there, isn't being released, then you have some issues, which is exactly what they were showing here. They didn't have the complete mechanism at the time, back in 2003, but Egan's paper was actually a really interesting one, demonstrating that the genotype is actually really important. I mentioned to you earlier that I could tell, even without having to genotype you, that all of you are actually probably homozygous or heterozygous for the methionine version. If you have made it this far to university and you have a certain memory capacity, even though you're here at U of T, so your decision-making skills may suck completely. But in terms of your learning and memory skills, they're fantastic. Because if you've made it to university, and you've made it so far in university, it suggests very strongly that you have these isoforms here on the left. So they wanted to take this one step further though. So BDNF, they knew at the time, was important for synaptic health. You can't really ask someone to donate their brain. Most people actually need their brains, so it's really hard for you to say, take my brain, go ahead and use it, return it whenever you're done. And so one of the things that they developed or were key in developing was this technique for looking at the effect of synaptic health. So if BDNF is really important for synaptic health, particularly in the brain area like the hippocampus, how can you actually go in and look at synapses back in 2003, 13 years ago, in terms of synaptic health? There is actually a technique. It's known as magnetic resonance spectroscopy. It's like a version of both the NMR spectroscopy that you learn in first year chemistry, as well as MRI. So you get a picture of your brain, and then you take a look at a particular area, and then you do spectrographs of different chemicals, and you can tell from the chemical spectrograph in a brain region, like N-acetyl aspartate, if your brain is healthy or not. And one of the things that they found was that in the brains of individuals who have one copy of the methionine, the marker for synaptic health, N-acetyl aspartate, using MRS was actually found to be lower in those individuals. Now, they didn't have a lot of data with the methionine. In a follow-up study, they were also able to show that the methionine subjects also had lower amounts of the NAA as well. And so this is the technique that I want you to be somewhat familiar with, this magnetic resonance spectroscopy. Sometimes it's abbreviated as proton magnetic resonance spectroscopy. And one of the things that it does is it can, in a brain area, using MRI, to examine molecules like NAA or N-acetyl aspartate, as well as other control molecules like creatinine that I'll show you on the next graph. And so you're looking at protons of chemicals in different regions of the brain. So if you're thinking about this, if you have nice healthy synapses because you have lots of really good BDNF out there, your levels of NAA should be really, really high in this type of spectrograph. And these spectrographs look a lot like this. You'll get an MRI done, so you can examine specific areas of the brain. And then shown here in this box, that is the area that you will actually go in and do a spectrograph of different chemical compounds that are found in that anatomical region. And when you look at all of the different proteins, protonated magnetic resonance profiles, this is the type of plot that you generate from this one little area. And what you're measuring is the overall size of this peak, the NAA or N-acetyl aspartate peak. And we use as a control either this creatinine peak over here on the right, on the left, as well as this choline peak. And both of these are a signature sort of peak. They're very, very characteristic. So we usually get a really big NAA peak, and then we get this double-shouldered peak here on the left. And we compare the ratio of this peak to this peak. And this is where we're talking about the normalization, because we're always comparing it to the size of this creatinine peak. No matter whose brain you're looking at, there are always individual variations. But overall, the variation for things like creatinine are very, very small. Most of you have a very, very small variation in terms of creatinine levels. It's also really easy to determine on a spectrograph. So when Egan's group did this, and they looked at the regions of the brain, like the hippocampus, after genotyping, individuals that had the methionine version of BDNF at position 66 had much lower levels of NAA compared to creatinine, suggesting that their synapses weren't as healthy, or there weren't as many synapses as they were in individuals that have valine-valine isoforms of BDNF. So again, I won't go into a lot of the details here. The main point that I want to make from this is the following. So we all have BDNF. In fact, you would not have a brain if you didn't have BDNF. The size of your brain might be a little different, or different areas of your brain might be a little bit different. But whether you have methionine or valine at position 66 isn't really going to affect the neuronal architecture too much. And so when scientists realized that there were these differences between the two different isoforms, when they introduced this into neurons, they actually saw no difference in the neurons. The neurons didn't suddenly become super huge, large neurons with tons of synapses with valine 66 versus methionine 66 having really tiny synapses or tiny dendrites. There were no changes in neurite outgrowth, the ability to make BDNF. None of that was affected by this isoform. The big difference is that if you have the methionine 66 version, you make it just fine. You make it as much as you make the valine 66. But remember, when we talk about synaptic plasticity, for something to stick in your hippocampal synapses, you actually have to release from neuron A that BDNF, and it has to interact with tract B. If you have methionine 66, you're going to make lots of BDNF. You're just not going to release it as easily. It's not going to be shooting out of your presynaptic terminal like a fire hydrant. It's going to be a slow drizzle instead of a large quantity coming out in a short period of time. And in fact, when you see the staining, most of the methionine 66 version tends to cluster around the cell body and doesn't really get released easily into the surrounding medium. So this is one of the reasons why we think these polymorphisms are incredibly important for us to look at, especially when we get into the brain, and especially in terms of learning, memory, and disease processes. And I wanted you to get an idea of why we're interested in genetics, why we're interested in this personalized sort of genomics that we're going to be getting into overall. So since that 2003 article, this single nucleotide polymorphism actually has been implicated in almost everything: anxiety disorders, anorexia, any kind of behavioral phenomenon that you can suggest has been looked at in terms of this methionine 66, valine 66 polymorphism. And again, lots of different papers. We won't go through other sort of papers related to this, but eating disorders, epileptic seizure activity, all of these different things have been implicated. So it is a really important, one small change actually in theory can lead to very big consequences in terms of a complex organism, a complex organ like the brain. Okay. Do you have any questions on that section before I go on? Or any questions about the techniques or any questions about the different isoforms of BDNF? Are you all clear on BDNF? So one suggestion I'm going to make to you, as you know, on your final exam, not that we're making it any more challenging or try to make the mark any lower, but I do want you to be able to demonstrate that you understand the material, not just from this lecture, but also to think about BDNF in the context of the lecture that we talked about with exercise. So in that lecture, we talked about how BDNF gets released in the brain, or what factors lead to BDNF being upregulated in the brain. So think about how you would tie in that lecture that we talked about with exercise and BDNF with this lecture that I've just given you right now. And that's sort of a big sort of heads up for you for one of the short answer questions that you'll get on the final exam. So this is a paper that actually came out a while ago. This paper has actually been really, really controversial. This is another polymorphism. It is not a single nucleotide polymorphism. It's not a change in a single nucleotide. This is actually a slightly different polymorphism in the genome. It's a change, but not at one single nucleotide level. This paper that came out also in 2003, oddly, is actually a paper that revolutionized the way that we look at major depressive disorder, anxiety, psychiatric conditions, and everything else. And this one paper, I remember being a graduate student at the time over at the Hospital for Sick Children. And this one paper caused such a really tremendous amount of excitement and interest. I remember people actually leaving the lab, running outside into the hallways and saying, did you read the new science paper that actually tries to explain depression? And it's sort of like the same sort of excitement that you might have over a brand new iPhone or something else. This is sort of the same excitement that scientists had when this paper came out. And I'm going to take you through a few slides related to depression, and I have to apologize. I see a slide coming up here that I didn't take out from the last lecture or series of lectures, or I might actually have the wrong lecture up here. Do I have the wrong lecture up here? I do? Okay, apologies. I think the USB key saved the wrong version of this. Can you tell me offhand which slide comes after this? Is it on depression? Is it a bar graph? Oh, great, thanks. Okay, I'll do it this way. I'll get a little teleprompter at the front. Apologies for those of you who are kind of watching at home. So depression is actually not a trivial disorder. If you take a look at the statistics for depression, it's actually more impactful in your life directly, especially for students. But I'm saying overall, for the rest of your life until you get to the age of retirement, it's going to impact your life tremendously. Much more so than cardiovascular disease, much more than diabetes. And you might think, yeah, you know, depression is never going to happen to me at all. Like my family's okay, I'm okay, I have no issues in terms of like depression. I'm a pretty happy, positive guy. But let me tell you that depression is going to impact you, and it's going to impact you at the worst possible time. It's going to impact you in this age range that, that we have between the ages of 15 to 44, and I think that's on your slide, correct? Somewhere on there, 15 to 44. Do you know why that age range is so important for you as young individuals? This is when depression strikes. Why is that age range so important? What's happening in your lives during the ages of 15 to 44? You're going out, you're getting married, you're starting a family, you're starting your jobs. This is supposed to be the most productive time in your life. You may not think so being here at U of T, it may not seem like it's the most productive time in your life, but the reality is this is actually the most important time in your life where you're really setting up your future and you're hoping for the best in your future. Now imagine during that time, you get sick where you have a broken leg that's going to take 2 years out of your life. During this time where you're getting married, finding a job and everything else. Or you get cancer during that time. And this is sort of the same thing. You're at a young age, you are not invulnerable to developing depression, which is why that slide is up there. And can I get the next slide? It's not that one. Sorry, and what is that saying? Epidemiology. Okay. So if you're looking at the epidemiology and the statistics that are associated with major depressive disorder, one of the things that you'll see is that it affects females more than males. It tends to strike in that age range that I mentioned to you earlier, and it affects a lot of individuals. It affects you a lot more than we actually have statistics that are available for you. Okay, I feel like I'm, okay, cool. Can I get the next slide?
So just go.
Oh, okay, thank you, wow. Wow, such a generous class. I've never had anyone give me a laptop before. This is really, really cool. This is one of the sad things about depression, and this is I feel dirty presenting this slide in 2016, and I do, because as a neurobiologist and as your prof, I should be telling you what we know about different disorders. The reality is that we know that it affects all of you. We know it affects individuals in this age range, but we actually don't know what the cause of depression is. There are some things that we know. That paper I showed you earlier at the outset, we know that if you're walking down the street, that suddenly you're not going to fall into depression. Usually, when you look at people with depression, they have a stressor in their life. It's something where someone in your family has passed away. You have health issues, you have a lot of anxiety. Whatever it is, there's usually a stressor that takes part in your life. We know that there might be a genetic component because there's often a linkage with family members that have another sort of mental health disorder. They might have bipolar disorder or BD, they might have major depressive disorder, but there's no guarantee. Even with identical twins, there's only an increased risk of developing depression at about 40 to 50%. There's no guarantee. If you have identical twins and they're put through the same things in life, there's no guarantee that both of them are going to have depression. One twin may and the other twin may not. So is there genetics associated with this? we're still not sure even to this day in 2016. I will tell you that it looks like there is a neurochemical change. As it says at the bottom of the slide, if you have one episode of major depressive disorder, you're more likely to have a second or third bout. You increase the likelihood from about 70% all the way up to, you're guaranteed, if you've had 2 events of major depression, you are guaranteed at almost 100%, 90%-ish, that you'll have a third bout of depression sometime in your life. So something is changing in your brain. Something in the neurochemistry is likely changing in your brain, although we don't know the exact mechanisms that are involved. How do we diagnose you with depression? And again, I don't need you to memorize this box. And one of the reasons why I put this up here for you, you should never self-diagnose yourself with major depressive disorder. You should always get it done. And these are the types of questions that a psychologist, a trained clinical psychologist, or a psychiatrist will actually take you through. One of the reasons why I'm bringing up major depression for you as a group, and some of you, again, you kind of poo-poo the fact that major depression not going to affect you at all. It's a lot like some of you thinking that I'm never going to catch a cold, I'm never going to break my leg, I'm never going to have this happening or that happening. When I started teaching 7 years ago in human biology, one of the things I really wanted to do was to impact my students and to really do something that made my classes memorable. And one of the things that I did was, in my very first class in 2010, in January, I stressed myself out for one whole month. Like for 6 weeks, I decided not to sleep for more than 4 hours a day, every single day for about 6 weeks, as well as eat irregularly. In other words, I was living the life of a U of T student for about 6 weeks' time. And the purpose of that at the time was really to see if I could stress myself out enough to actually induce major depression. So I finished all of that. I finished this whole regimen. I slept for about a day and a half after I finished all of that, and I came back, I walked into class. I was so arrogant at the time, I slammed my laser pointer down at the front podium over in Wetmore Hall, and I said, see, I can do it, I'm not depressed, so why would students have major depression or other mental health disorders? The big issue is 6 months later, after that was done, one of the things I realized is I had some of those symptoms. I had things where I was no longer motivated, no longer interested. There were problems with the way in which I slept. There were problems in all sorts of different cognitive aspects. I couldn't concentrate as well as I used to be able to. And one of my colleagues in the office, she suggested at the time, you know, I think you might have depression. You should go get yourself checked out. And I had the same response that many of you had. You know, I'm not depressed. It's just I'm really tired or something. But the reality is I did have depression, and it did last for about a year or so, about 9 months. And I won't go through all of the details now because we're running a little bit short of time. We don't know the mechanisms by which, for example, my brain changed. We do know a couple of things, that there are some neurotransmitters like serotonin, noradrenaline, and dopamine that are implicated in those different sorts of effects that we just saw there. Well, why is that paper that I showed you earlier, why was that paper so exciting for us way back then in 2003 as neurobiologists? That paper in 2003 brought all of the theories about depression together. So you know that you can't get depression unless you are stressed. So it brought in this element of stressors. It brought in the element of genetics, because we're not clear on the genetics, but there is some linkage to the genetics. And it brought in this concept that maybe one of those different molecules, a neurotransmitter like serotonin, was somehow implicated in this whole process. In that paper that I showed you earlier, I said that this is not a SNP. And what they identified in this cohort of New Zealand individuals, they did a prospective study. They decided, you know what we're going to do is we're going to take about 800 individuals. We're going to see how they're doing today, and we're going to follow these young individuals as they get into teenage life, into adulthood, and we're going to do this long 15-year study. And then we're going to see how many of these individuals get depressed during that time. And when they did this, And they saw that there were clear differences. If there was a stressor in the life of an individual that they started looking at over this long prospective study, they realized that there were differences in this particular gene that is encoded here on chromosome 17 in this serotonin transporter. And this serotonin transporter has a long version or a short version. Are you guys all on that same slide? I know you're not on that slide, but oh, it's on your phone. Okay, cool. And one of the things that they found was that it actually did not encode in the coding region for the serotonin transporter at all. What it encoded for was in the 5' region of the variable nucleotide tandem repeat, or the 5' VNTR region of the gene. Now, think a little bit about this. What do you know in terms of biology for the 5' VNTR? What does it do? The variable nucleotide tandem repeats. So those are used as promoter regions. So individuals that have the short version actually have the same size transporter as people that have the long version. They just don't make as many transporters as individuals that have the long version. So short isoforms means that the promoter isn't turned on as much. It means that the transporter doesn't get transcribed nor translated as much, and as a result of this, it means that these individuals actually have less serotonin around, and if they are stressed out for whatever reasons, and they happen to have that genotype where they have the short isoform of the serotonin transporter, they are more likely to fall into depression. It's just a probability, it's not a guarantee, but they were able to show in that Caspi paper in 2003 that stress with this 5-HTT-PLR short isoform gave you a stronger likelihood of developing depression during your lifetime if you had this genetic polymorphism. It was a remarkable paper, but like most of the papers that we've looked at, it has fallen into a lot of controversy. There have been no fewer than 16 follow-up papers based on this one original article back in 2003. Every year, someone else's group comes out and says they agree, and every year someone else's group comes out and say they disagree with this. Currently, we're at the point where we think that this data is actually correct. So again, I have to apologize. The last few slides are actually very similar, and I'm probably going to leave those to the lecture on Monday. So for the weekend, there are a couple of things that I'd like you to make sure that you're doing. Some of you have already started preparing, and just as a show of hands, if you had to write the final exam on Monday, how would you do? 0. Is that 0 or okay? I'm not sure. Right now, I'm going to ask you to do two things. I'm going to ask you to actually go backwards. I don't want you to be going and studying all this stuff from a while ago, unless you have been throughout the semester. I want you to start from today's lecture, work your way backwards, spend some time on CRISPR. You know that there's going to be a lot on CRISPR on the final exam. Spend time on that over this weekend. I want you to go to tutorial. I know that the temptation is not to go to tutorial, but go to tutorial, try the practice questions. You'll have some indication as to how you're going to be doing. The questions will be of similar types that you'll get on your final exam. I do want you, right now the class average is really good. I like the class average. I don't know if you like your class average. I like it. It's about a B+-ish, which is not bad for a second-year class. I'd like it to stay at least at a B+-ish range. And I'm not intending to make the final exam harder in order to try to drop the average in any way. I just want you to know that there's a lot of material on the final exam, and that some of the questions are not trivial unless you've actually studied it and unless you've actually spent time on it. So please take time over the weekend. Think about this, and as I mentioned to you today, from today's lecture in particular, go back to the exercise and BDNF lecture, tie those two things in. BDNF today and BDNF from exercise. Figure out how you'd write a short answer question related to that on the final exam, and that will be one of your short answer exam questions. Okay, and have a good weekend. Hopefully next week the slides will be back in order. Sorry. Thank you so much for that. I appreciate it. Thank you.